Let's talk about transcription. Now this whole unit really revolves around protein synthesis. Now you've learned in previous science courses maybe that a gene is a section of DNA that codes for a specific trait. And that's true, but if we're being a little bit more specific, we're going to say that a gene is a section of DNA that codes for the making of a specific protein. And it's the protein that in a number of ways would cause you to have that trait. So that's what this whole unit revolves around, protein synthesis. And there's two steps in this whole thing. Step number one is transcription. Oops. And we're going to talk about that today. And the second step is called translation, and that's a topic for another video. Anyway, overall, the protein synthesis goes like this. We use the DNA in the cell to make something called mRNA. RNA, as you know, is a chemical very similar to DNA, but it's a few differences. For one, it's single-stranded. And it's mRNA, it stands for messenger RNA, because basically it's a message from the DNA on how to make the protein. So that's step one. I'm taking the DNA in the cell and I'm using it to make a strand of mRNA. And that's called transcription. So that's all we're doing. And basically it looks like this. Now this transcription thing that we're talking about, using the DNA to make a strand of RNA, it requires an enzyme. And that enzyme is called RNA polymerase. As you know, enzymes always end in A's, right? And so a polymerase refers to an enzyme that's making polymers, or in other words, building a bigger thing. So RNA polymerase, basically, as the name suggests, builds RNA. So here's the enzyme, RNA polymerase. And here's my double-stranded DNA. And what it does is it looks for a promoter region. And the promoter region is just a series of T's and A's in the DNA molecule that says, basically, it's telling the enzyme, start transcription here. That's basically what it does. So when it sees that promoter, the enzyme is then going to do two things. Number one, it's going to separate the two DNA strands in one little section. Okay? And that's called a transcription bubble. And when it does that, it's then going to use one of the DNA strands, just one, and it's going to make a complementary strand of RNA. So remember what I mean by complementary. A is complementary to T, A always goes with T, and G is complementary to C, like G always goes with C. The only difference here is that in RNA, I never have T's. Wherever I expect to see a T in RNA, I have a U instead. U stands for uracil. Alright, so very basically, if this is the DNA strand that the enzyme is using, it will just move down the DNA strand and it will use it to make a chain of complementary RNA. And then when it gets to the end, you'll see a stop sequence of sorts. The enzyme will detach and it will release my RNA. Okay, so I have made a strand of RNA that is complementary to the DNA strand. In other words, if this is my DNA strand, TAC, GGA, blah, 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 etc., I have made a strand of RNA that is complementary. So instead of T, there's an A. Instead of A, there would be, well, you'd expect the T, but because this is RNA, it would be a U. And C goes with G, and G goes with C, etc., etc., etc. So I have made a section of RNA that is complementary to the DNA. And that's all that transcription is. So let's take a look at a little video here. Come on. Hopefully it doesn't take too long to load up. Nice, okay. Let's make that a bit bigger. Okay. So here's my double-stranded DNA, and it's made of A's, T's, G's, and C's, as you know. And you know, if I've got A over here, then this would be a T, and if I have a C, this would be a G. You know, it's complementary. Okay. And first thing that the enzyme does, RNA polymerase, is it's going to separate the two DNA strands. Okay, so there it is. Separates the two DNA strands to make a transcription bubble. And what it's going to do is it's going to start adding complementary RNA nucleotides to one of the DNA strands. It's going to make a complementary strand. So there's the first one. And if this was a G on the DNA, then this would be a C on the RNA, of course. And it's just going to move down the DNA, and as it does, it's going to continue to add complementary RNA nucleotides. So it just moves down the chain, adding the complementary sequence of RNA. Notice again that the RNA that we're making is single-stranded, and that's one characteristic of RNA that's similar to, dissimilar, not the same as DNA. And then when it's done, it just releases the RNA, and we're done transcription.